And you can please ask questions all the time, and you do that in the chat. And if it is questions that we need to answer to understand the topic, then I will interrupt Dan and ask them when it's suited. And otherwise, if it's more about the discussion, we will take it in the end of the meeting. And I will also want to say a big thank you to all of our partners who are making this week possible and free for everyone to attend. So thank you very much for that. Without you, this wouldn't be possible. And I also want to say a little bit something about why we're organizing a soil health festival. And I told Dan earlier that we actually got really inspired by the work that they have been doing and having this 16-week online soil health conference in the US. And then we thought that, okay, we have to make something similar, a little bit smaller, a little bit more packed, but we actually got really inspired by that. Because we at Svensk Kolinlagring, we think it's really, really important to spread the knowledge about soil health and how important it is for the transformation of our society to become more resilient and sustainable. And we all know the climate change is happening here now, and our soils also have to be in good shape in order to tackle these the climatic changes that we are facing. And the way that we are producing and consuming food today is the biggest driver of the biggest negative driver of oh shit, what's that? Yeah, the loss of biodiversity and freshwater use, change of how we use the soil, etc. So our food system needs to change dramatically, and therefore we also we look at this topic from a lot of different angles during this week. And as also like a thread through the week, we also talk about who is actually responsible for that this change will actually happen. And tonight we're going to talk about soil health and nutrient density, which I think is super interesting. I really, really believe that nutrient density could be the leverage that can transform the food system a lot faster in a way that we want it to change. So I warmly give the word to you, Dan, to guide us through this topic, and we will have time for discussion in the end as well. So while I'm finding your presentation, Dan, you can just introduce yourself.
Okay, great. Well, thank you for having me. I'm excited to present to you. And yes, my Swedish is non-existent, so thank you for letting me speak in my native tongue. I'll try to speak slowly. I'm not quite sure what the level of comfort in English is. I have a habit of speaking too fast sometimes, so I'll attempt not to speak too rapidly. My name is Dan Kittredge. I'm the founder and executive director of the Bionutrient Food Association. We're an educational nonprofit that we've been around for about 15 years. So I'm going to walk you through here in this slide presentation some of the work we've been doing on this topic of nutrient density. But maybe I'll just start with a quick background on myself. This graphic here is not only the front page on our website, but it's also a drone shot of the farm I grew up on. So I have a personal connection to farming, smallholder farming. Sort of grew up on a mixed farm where we had cows and pigs and chickens and ducks and geese and orchards and vegetables, and it was a homestead, back to the land homestead. So it was a broad suite of production. A lot of it was for personal consumption originally, and then it's now turned into a more of a CSA farm and things. My parents also were actively involved in the organic movement. Helped write some of the first certified organic standards in the US in the 80s. Ran a local organic farming organization for 30 years here in Massachusetts as part of their professional career. So I come from that background. And uh, when I got married and had found no better way to raise a family, I thought, than on the land as well. Set about trying to be a farmer, and that's when I realized that I was, like a lot of farmers, um, not good enough to make a living. We had struggled with issues of pest pressure and disease pressure and failure to thrive. And even though I was well versed in the organic orthodoxy, I didn't necessarily understand how nature had evolved things to work. How plants have been growing for hundreds of millions of years just fine without fertilizer and tillage and chemicals. So I set about reading books and going to conferences and attending seminars, and um, broadened my base of understanding and took sort of more some nuances home of different production practices that were able to help me experience, pests no longer being able to eat my plants, diseases no longer being able to eat my plants, yields increasing, um, shelf life, flavor, aroma increasing. And I was able, after not too much effort, to make a living working 20 hours a week farming. And I thought at that point in time that perhaps there was something important here that could be shared, and have started this educational nonprofit, the Bionutrient Food Association, in 2010, with that intention of sharing the work broadly. But part of what I had come across when I was doing my research was this concept, a term called bricks. It's a measurement scale like Richter or Fahrenheit or centigrade. And there's a simple instrument you can use called a refractometer, and you can squish a carrot or a potato or a tomato, and get a drop of juice out of it, and you can look at it through the refractometer, and it'll tell you relatively how nutritious it is. And this concept was basically that there was dramatic nutrient variation in food. And Yeah, that was a revelation to me, that not all carrots are uniform, not all potatoes are uniform, not all chicken is uniform, etc. There's dramatic nutrient variation in food depending on, it seems, how it was grown. And so um, that connects to flavor, aroma, human health, soil health, it seems like, in a very positive fashion. So um, we coined this term in 2007, 2008, nutrient density, to refer to that variation in nutrient levels in food, as in this carrot is more nutrient dense than that carrot. And so what I want to proceed through here in this presentation is the work we've been doing since coining the term, the research we've been doing, the findings we've come up with, the instrumentation, the opportunity to use nutrient density as a vector to inspire the supply chain or agriculture broadly to be much more ecologically beneficial, which we think would also cause it to be much more, um, you know, beneficial for human health. So if you want to just go to the next slide, Jessica. Yeah, the Bionutrient Food Association, that's the name of our organization, we were founded in 2010, and our mission is to increase quality in the food supply. And by quality, we're talking about flavor, aroma, and nutritive value. And so basically, yeah, we would like the ambient level of nutrition in food next year for the people on the planet to be higher than it is this year, and proceed forward in that direction. There has been um, a decrease in nutrient levels in food now for quite some time, that's been documented by various governmental agencies, and it does seem that that is correlates quite well with increases in chronic disease, etc. So next slide. So we were, in the first few years, we did a lot of educational work, courses. Primarily, I would teach courses to other farmers about what I call principles of biological systems, how nature evolved things to work, how it is, as I said, that plants have been growing for 400 million years or more, just fine without tractors and fertilizers and things. And we set up an annual conference. We had local chapters. Some of these people here you see are sort of leaders in the broader movement, that we've had speak at our conference. So as the sort of the work spread and other farmers were having the experiences I was having, which was that their cost of production was going down and their pest and disease pressure was was decreasing and their economic viability was improving, would sort of tell their friends, and then we'd get invited to go give a course in this part of the country or that part of the country, and sort of spread through word of mouth further and further afield, the sort of more integrated perspective of agriculture. And as we began to see that it didn't really seem to matter what your soil type was or your climate zone, what the scale was, what the crop was that you're producing, that these principles worked across the board, we started to think about how could we take this and bring it to a larger scale. So can we just go to the next slide? So we started in 2016, sort of conceptually putting together a strategy for research. And we had effectively three basic questions. The first question was, what is the nutritional variation in food? We surmised that it was significant, but there wasn't actually an established database that sort of verified that spectrum of variation. Our second question was, if nutrient variation was dramatic, what caused it? People who we were talking to, who are sort of professional researchers, were saying it's probably not that big, but if it is, whatever variation does exist, probably connects to variety or soil type, and those are likely going to be your factors. Our thought was, and this was starting, before regenerative was really a word, that there could be a very powerfully positive opportunity for agriculture to have, beneficial ecological impact. And so one question was, if nutrient variation is dramatic, is it caused by things like soil health, or is it really back to genetics and soil type? And the third question was, is it possible to build a handheld consumer priced flash of light meter, effectively a ray gun, where you could walk into a grocery store or a farmer's market or anywhere you would be choosing food, and with a flash of light determine from the options on the shelf which ones were more and less nutritious. So imagine there's three different brands of carrots in your grocery store, and you can flash a light, 20 out of 100 on the first, and 40 out of 100, and then 80 out of 100 on the third. Our thought is people who are buying food for themselves and their families to be well would probably choose the food that's more nutritious and leave the less nutritious food on the shelf. And our thought is that could be a very powerful economic driver to incentivize farmers to focus on nutrition as opposed to volume. And it seems like it's that focus on volume which has caused a lot of the detrimental impacts environmentally. So basically using economics as an inspiration to shift the way farming is done, that was the vision. So we put that together in 2016, and we had these three questions: is variation dramatic? What causes it? And can meters be built to test it? I guess we can just go to the next slide here. We started in 2017. We built our first meter.
Just can you just show us, describe what we are seeing here? It says grape 15 times and zucchini 5 times. What does it mean?
Yeah, so there actually is on other versions of this graphic, there's a bunch of text in the middle which explains it. But so we did proceed forward to do a lot of research. We sampled a total of more than almost 10,000 samples of crop from 25 different crops over multiple years. And by averaging those, I think we did 6 elements and 2 compounds, so copper and zinc and phosphorus and potassium and polyphenols and antioxidants. And we looked, if you take the carrot with the lowest level of all those and the carrot with the highest level, what was the range of variation? In this case, it was 3x. In winter squash, it was 4x. In zucchini, it was 5x. So that would mean that this carrot has as many nutrients in it as those 4 carrots, or as many nutrients, or the zucchini as these 5 zucchinis. Or in the grape, this one grape has as many of these 8 nutrients in it as 15 other grapes. So that was the overarching scale of variation we found across the board. But yeah, I was going to walk through the steps of what we did each of the years to explain the science behind it. Effectively, the first step we took in 2018, well, in 2017, we built our first handheld meter. And then in 2018, we built our first lab to begin assessing crops. We had people that year send in just carrots and spinach and look at the we looked at, again, like about 12 different elements. We wanted crops from grocery stores and farmers markets and all parts of the country, organic, not organic. We were surveying the supply chain to see what the nutritional variation was in the supply chain. In 2019, we set up our second lab out in California, and we began to have farmers who submitted crops send in the soil those crops grew in, as well as answer a bunch of management questions like, what was the variety? Where did you get it? How did you prepare the soil? What were the fertility programs you used, et cetera, et cetera. And yeah, let's just go to the next slide, and we
Yes.
can look a bit more in detail here.
I'll just see if I can. Yeah.
Okay, so yeah, 2020. This is our third lab year. We set up our third lab in Europe, in France, and if you can see, all of those sort of green icons are farmers, and those farmers were sending in the crops, sending in the soil they grew in, and sending in answering a bunch of management questions, so we can begin to overlay the nutrient levels in the crops against the dynamics in the soil, against the environmental conditions, causal factors. You can see on the right the number of samples of each of those. We got 398 carrot samples that year, and 118 bok choy samples, and 89 grape samples, et cetera. Um, on the bottom left, you can see what the nutrients were that we were assessing in the crops, and then the center, the aspects of the soil that were being measured. So this is it's a preliminary work, but we wanted to sort of begin to populate a database to see what the scale of nutritional variation in food was, and these are the things we're looking at. So let's just go to the next slide and look at some of the things we found. So again, these are slides. These next 3 slides will be just having to do with carrots. I had to choose one crop out of the many crops that we did. Understand that the things I'm about to explain are very similar across the board with all the other crops we assessed. More than, I've got graphs like this for more than 20 other crops. If anyone wants to go look at our data, bionutrientinstitute.org is the website where we have all this housed. We have our reports. We have a data explorer. You can go and look at the data directly, see the papers that got published, et cetera. So on the left, you can see we're looking at sulfur in carrot. You can see on the bottom left-hand corner, it says 8.41, and on the right, it's 33.19. Those are milligrams of sulfur per 100 grams of fresh weight. So that's just the amount that was present in those carrots. So 33.19, you divide that by 8.41, and you get about 4, which basically means that the carrots on the right had 4 times as much sulfur in them as the carrots on the left. And then with the fifths of the range, the red, orange, yellow, green, light green, dark green, and if the 8.41 on the bottom left was 0 out of 100, and the 33.19 was 100, then that where the red is basically 0 to 20, the orange is 20 to 40, the yellow is 40 to 60, the light green is 60 to 80. So you can begin to see the how many samples were where in the continuum. If you see it's tallest there at the beginning of the orange, what we would say is like the 20th to the 25th percentile. So what we can say is a vast majority of the samples were below the 50th percentile of what's possible. We identified a range of sulfur that existed in carrots, which was 4x, and the vast majority of the samples we assessed were at in the bottom third, effectively, of what was possible. On the right-hand side, you see phosphorus, and that's from 11.7 is the lowest we found to 93.69 is the highest we found. That's an 8x variation, an 8 times variation. So there's 8 times as much phosphorus in the carrots on the right as there were in the carrots on the left. Again, the peak of the curve is in that, below the 30th percentile. So we have some in the top 20th and top 40th percentile, but the vast majority are not. When I'm presenting this, I like to say, when I went to college, to university, we had a grading system, which was that any final score above 90 was considered to be an A. Anything between 80 and 89 was a B. Anything between 70 and 79 was a C. Anything between 60 and 69 was a D. And everything below 60 was an F. And that was a failure. You'd fail the class if you had your final score was below 60 out of 100. And in this case, all of the red, orange, and yellow is below 60. Right. So what we're seeing here is if farmers were graded on that scale of what's possible versus, what they get from what's possible to have in the crop versus, what was in the crop they produced, the vast majority of them would be effectively getting failing grades. So this is just the nature of the supply chain that we've been able to assess. We can go to the next slide if you want, Jessica. These
Yeah,
next
we
two.
have some questions also in the chat.
Okay.
Because now we're looking here at antioxidants and polyphenols as well. So one is if all separate nutrient levels given the same weight when calculating a composite level, or if not, how do you do about that? So when you, yeah.
So we are yet to come to the point where we think we can say this is good, this is bad. Right now, what we're doing is we're assessing the scale of variation to understand how, are carrots relatively uniform, or is there a dramatic nutrient variation in carrots? And I'm not sure what it's like in Europe, but I know in the US, if you were to buy a bag of carrots, oftentimes there'll be a little a nutrition facts panel on the side of it, which will say, you know, so much vitamin D per 100 grams, so much calcium per 100 grams, so much potassium per 100 grams. They'll lay out on the side of the package what the nutrients are in those carrots, and that does not come from actually testing the carrots. That comes from every 10 or 20 years, they'll take 10 or 20 samples of carrots, find an average out of the carrots that they've assessed, and presume that that is what is in all carrots. And so our foundational point here is to say, look, all carrots are not nutritionally uniform. All, wheat and oats and potatoes and lettuce and tomatoes and grapes are not nutritionally uniform. There is a dramatic nutrient variation, and
Yeah.
there has not been a database created yet to really characterize that. So let's begin the process by doing this data collection.
But I think what Gunnar is asking is also like, because you have these different compounds that you are measuring, and then together they can get also, like now it's for each and every compound, they can get a score from 1 to 100. But then you can also get a score from 1 to 100 when all of these are packed together, right? So how is the different, how are the different micronutrients weighed in that when you put all of that together in the final score from 1 to 100?
Yeah. That's will be the second half of this presentation. So if you just wait about 15 minutes, hopefully that answer will become clear.
Yes.
What I'm walking through is effectively what we've done and what we found to sort of end up with that conclusion about what to do now. But the first point is, the first question when we started in 2017 was, is there dramatic nutrient variation in food? Because all of the researchers and things that we went and talked to said, no, there wasn't. It was maybe 2%, maybe 5%, maximum possibly 10% was the variation top to bottom of nutrients in food. And so this process, for the first thing we need to do is sort of assess whether that's true or not. And to do that, we engage in sort of a epidemiological style of analysis where we're, broadly assessing the environment, not doing replicated randomized trials. So, yeah, on antioxidants here, which are understood to be, very health beneficial. We can see that the lowest level we found in carrots was 4.92 FRAP units per 100 grams of fresh weight, and the highest was 195. And that is a more than 40x variation. So the carrots on the right had 40 times as much antioxidants as the ones on the left, 40, right? You'd have to eat 40 of those carrots on the left to get as many antioxidants as the ones on the right. And again, the peak of the curve there is in the the 15th percentile. On the right is polyphenols. Polyphenols are compounds that are very complex. They correlate with flavor and aroma, at being sort of anti-cancer, anti-diabetes, anti-heart disease, etc. Beneficial in relation to, reducing chronic disease, for instance. And the variation there was from 2.34 to 49.75. So a dramatic, that's 20x variation, and the peak of the curves there is below the 10th percentile. So
We have another question here from the chat then, if it's possible to get dangerous levels of certain nutrients.
yeah, well, these are all nutrients that were understood to be deficient in people consuming a quote unquote normal Western diet. These are health necessary, and the general populace is, according to medical practitioners, deficient in these nutrients. So certainly excess is possible and detrimental. I think what we're sort of pointing out here is that food in general, as it's being produced right now and is available to consumers right now, is relatively quite low in relation to what its potential is, which in many cases then correlates to overeating, and obesity and other things, because people's bodies need these nutrients, and even if they're eating calories, those calories are relatively empty from a nutritional standpoint. But yeah, let's proceed forward to the next slide so we don't spend too much time on this preliminary stuff. I want to try to get it done. So here we are looking at the some of the what may be the causal factors in this variation. Again, a lot of the researchers were telling us that they thought that variety was a major piece of the puzzle. So you can see Napoli, Bolero, Nantes, Mokum, Romance are all different varieties of carrot, and we can see that you can plant that seed in one soil and grow it one way, and they'll have low levels of nutrients, and the different soil, high levels of nutrients. We're not seeing any correlation there between variety and nutrient level. On the right-hand side, you've got various different, they're called climate regions by the United States Department of Agriculture: Northeast, East, North Central, Southeast, Central, Northwest. So different soil types, different climate dynamics. Again, we don't see any correlation between where the crop was produced and what's in it. Let's go through the next slide. On the left, we've got by sample source. So where were the carrots sourced from? On the top, we've got farm. On the middle, we've got store. The next one is garden, and the bottom one is farmer's market. So again, it doesn't seem like you can predict relative nutritional caliber in food based on where you purchase it from, or whether you grow it yourself. In most cases, it's nowhere near what it could be, is what we're finding anywhere we look in the supply chain. On the right-hand side, on the top there, you see certified organic. And so again, a lot more red than there is dark green. Plenty of oranges, which is in the 20 to 40 range, but not a lot of those in the dark green. Cover crops, people who use cover crops is the next one. No-till is the one after that. Tillage, organic, not certified. Reference and none there are the ones that are from grocery stores and not organic. Regenerative is the one below that. Biodynamic is the next. Local, there is the sort of the one that actually is most evenly spread and fattest over towards the green. But again, we're not seeing any correlation between a label or an individual practice or a certification and the relative nutritional value of the food. It seems to be that generally across the board, what's in the supply chain is relatively low in relationship to what it could be, and there's dramatic variation. The only thing that we found that did connect with increases in nutrient levels was increased levels of life in the soil. So this is something that, for those of us who understand evolutionary biology and how plants were effectively evolved by microbes 420 million years ago, you understand that only when a plant has a well-functioning gut flora is it healthy. It was the microbes that evolved the plants, and so it does make sense that there would be a connection between the health of the plant and the level of vitality in its microbiome.
We have a
Okay.
question here as well, Dan, like from the chat, like if the nutrients drastically decrease from the time when they are harvested, and is this something taken into account?
Well, the one thing that we controlled for was we said we want all of these crops to be sourced from where the person would be able to buy them and eat them. So off the market shelf, wherever that was, from a CSA bag from your local farm, from your grocery store, from the farmer's market, we sourced the crops from the point of purchase. So I think the real point is there's a dramatic variation in what's on the shelf. A lot of these nutrients, like copper and zinc and phosphorus and potassium, these elements, they do not degrade over time, right? That carrot can sit there, it can rot, it can dry up. It'll have the same amount of calcium in it 2 months later as it had when you started off. So certain compounds will degrade, like the polyphenols and antioxidants, they will break down over time. But a lot of those basic elements absolutely don't go anywhere. So yeah.
I was just to stay a little bit with what actually was a correlation with the soil health. So
Yeah.
just want to say again, how do you measure that? Like how was that
Yeah.
done?
In this original level of our work, which we used a method called respiration. So we would take the farmers would send in the soil that the crops grew in, and we would dry it out in an oven, and then we would take a certain amount, measure it out, and take some deionized water and measure it out, put the soil and the water into a container, put a lid on it, and then measure the amount of carbon dioxide that was emitted um, after 12 hours and after 24 hours. So because a lot of these microbes in the soil are aerobic, they breathe in oxygen and breathe out carbon dioxide. The amount of carbon dioxide emission is a good proxy for level of soil life. It's not a sophisticated DNA analysis, but it is is understood to correlate very nicely. As a nonprofit educational organization, in this original phase of our research work, we were not well funded, and so didn't have the budget to be doing more complex, um, um, individual species identifications.
Yeah. So last question here, if you looked at frozen products as well, or only fresh produce?
This was only fresh.
Yeah.
This is only fresh. And we would assume that if something was picked ripe and frozen, that it might maintain a lot of those nutritional compounds that could degrade over time. So if you take, you pick 2 pints of blueberries and you freeze one and you take the other one and you put it on a truck and drive it, and it sits on a shelf and you eat it a week and a half after it was picked, um, the understanding would be that something that was picked ripe and frozen would have higher levels of nutrients in it than something that's sat for a week and a half. But we did not look at frozen products. We only looked at fresh. Yeah.
Yeah. Thank you.
Cool. All right, let's go to the next slide. All right, so this is and by the sort of middle end of 2021, we were able to release the second generation of our handheld spectrometer. So I've got one here. I think Jessica has one as well. It's a fairly simple, fairly, rudimentary thing, but it is a functioning handheld flash of light meter. You put the carrot on this screen right here, or the lettuce leaf, or the wheat berries, and through the app on the phone, you cause it to flash the lights, and it takes a picture of the light that bounces back and gives you readings. The antioxidant level in this carrot is here in the scale of 1 to 100 of what we found. The polyphenol level in the zucchini is there. And yeah, so we were able to, by 2021, answer our 3 questions in the affirmative. Nutrient variation is dramatic. It does seem to correlate with soil health and not with soil type or variety. And it is possible to build handheld consumer priced meters that could be used at point of purchase to assess that variation. So with that exciting sort of conclusion, we begin our next step of the work, which is these last few slides here in defining nutrient density. So yeah, you want to move on to the next slide? Great. So so we'd identified that nutrient variation was dramatic, but that was entirely different than saying what is good, what is bad, to one of the earlier questions. To be able to say that this carrot is 80 out of 100 or that carrot is 20 out of 100 requires a much higher level of complex science than we had originally done. And so we have started, we're almost complete now with the beef project. We chose beef as our first crop to define nutrient density in because it has a massive ecological footprint. Many acres or hectares on this planet are used in the production of beef more than any other crop, foodstuff. It also has a very large economic footprint. A lot of money is spent on the purchase of beef. So if we're going to try to use this awareness around nutritional levels in food to shift the way farming is done to have positive impacts, we thought we should focus on something that's got a bigger footprint than smaller because it's an expensive process. So our thesis here is that effectively, if cows are eating what they evolved to eat, their milk and meat will be more nutritious than if they eat the grain that they've been fed now to greater and greater degrees in the last 100 years. So let's move on to the next slide. So the process by which we're doing this is farmers are sending in samples of the meat, the steaks, a steak from 3 different animals on their farm. We started off looking at about 800 different nutrients in those steaks, identified which ones were varied and which ones didn't, and then now in the sort of the final phases, we've been looking at about 100 different nutrients in each steak, from elements to vitamins to enzymes to fatty acids to polyphenols and terpenoids. So a broad suite of nutrients in the meat we were looking at. The farmers are also sending in manure, so we're doing a microbiome assay on the manure to see what the species are in the gut of the animal, which we understand connects well to animal welfare, animal health. The farmers are sending in the forage, whatever it is the animals are eating, whether it's a grain ration or fresh green grass or hay. We're assessing that. They're sending in the soil that that food grew in. We're asking a bunch of questions about the animals, like what was their variety, gender, age, how they were raised. Were they spend the last 6 months in a feedlot, or were they out on grass, moving every 2 days, or what was the sort of how they were raised? And then we've also done some human health trials. So feed this meat to people for a week and see what happens to their inflammation markers in their blood and urine, and then feed that meat to the same people and see what happens to their inflammation markers. So the idea here is by overlaying the data from the biochemistry, from the nutrients against the microbiome, against the forage and soil, against human health outcomes, we can find patterns which will give us the ability to say, okay, these 5 nutrients can be used as a calibration mechanism for a meter to be developed that can give that answer of that 80 or 40 or 20 out of 100. So we don't know what the answer is. We will be able to find patterns, we think, by looking at all these things in relationship to each other. So that's the general strategy for developing nutrient density, for defining it. Next slide. Well, yeah, you can see the geographic diversity of the samples. We actually have a few more there in Australia than is recognized on this graphic, but samples from 4 continents and a broad diversity of ecological environments. Next slide. Some of the preliminary findings, people may have heard of this thing called omega-3, omega-6, omega-6, omega-3. There's different kinds of fatty acids, and they are correlated to inflammation in people, depending on what the ratios are in your body of omega-3, omega-6. And inflammation is very foundational in chronic disease and the cancers and diabetes and heart disease and stuff like that. So you want lower levels of inflammation in your body, and this, the more omega-6 you have in your body, the higher the level of inflammation is. So I think the American Heart Association tells us that any food that has a greater than 4-to-1 omega-6 to omega-3 ratio is bad for you. I think it's, researchers who've looked back into traditional diets of humans for thousands of years say that we generally had a diet of less than 2.5-to-1. So 2-to-1, 1.5-to-1, that was the range of the ratio of omega-3, omega-6 that we evolved in. And you can see here that the meat from the cows who ate grass at the end of their life, or all their life, the average ratio was 1.7-to-1. And the average ratio on cows that were eating grain at the end of their life was 7.8-to-1. So while this is not a comprehensive definition of nutrient density, it certainly is a one compound ratio that is well published, that is understood to very nicely connect to human health. And we can see dramatic variations here in the omega-3, omega-6 ratio in the meat of the animals based on what it was they were eating. Okay, go to the next slide. Yeah, this is my last slide here. And this basically is just showing that the omega-6, omega-3 ratio is, in the meat does correlate very nicely with the number of species of plants that animal had to eat. So over there on the left is the corn, basically. Very, very small levels of diversity in the diet. And then as it proceeds over, the omega-6 3 ratio gets much better as the diversity of plants that the cow has access to eat increases. So at least preliminarily, our findings seem to be supporting that concept that if cows eat what they evolved to eat, they're healthier and better for us to eat. But again, with the industrialization of agriculture, in many cases, what we have available to us on the shelves has high levels of these anti-nutrients or health detrimental compounds. It's not just a question of high levels of good things, it's also a question of high levels of bad things. We're not talking about toxins, chemicals and pesticides and herbicides. We're talking about the actual thing that the food is made up of, which in many cases, like we're showing here, how that animal is fed can have negative implications. It effectively is, to broaden this out a little bit, it's grains and seeds that have high levels of omega-6s and green leafy things that have high levels of omega-3s. And so you could fairly easily make a jump here and say that whether a cow is eating a lot of grain, or a pig is eating a lot of grain, or a chicken is eating a lot of grain, all of those animals, whether it's the milk or the meat or the eggs, would have unhealthy levels of this omega-6 in them. This is not what animals traditionally have eaten. This is what they've been fed now, with the industrialized food system that can be more mechanized and mass produced, but it is not healthy for the animals, and it is not healthy for humans. So yeah, right now we're at the phase of working to establish what we're calling the Bionutrient Definition Standards Board. So we're welcoming microbiologists to join the board of people who look at the microbiome data, nutritionists look at the nutrition data, biochemists look at the biochemistry data, and then have them come back to us and tell us whether they think, yeah, which animal had the best gut flora, which animal had the best nutrient profile, which meat had the best human health outcomes, what were the best ecological dynamics for those animals to be raised in. And if we find patterns, then we'll be able to calibrate next generation instruments. I've got an example of a sort of a simpler, well, actually it's more complex, but less expensive, more mass produced meter here. This is it's called a magic pencil. It's just mass produced in China, and it costs a lot less than the thing we made, and it's a lot more sophisticated. And there's lots and lots of these meters out there that can be used when we have definitions of quality to assess it. And our thought is, if people have the ability to inexpensively, simply choose off the shelf the things that are better for them and their families, and leave the things that are less good on the shelf, that'll provide the economic incentive for this broad improvement in the food supply to occur. Our vision is that in the end game, the camera in your smartphone can do this. You need not hold a separate spectrometer. You can literally open up an app and take your camera, your phone's camera, and flash a light at the carrot or at the beef or at the milk, and be able to know nutritionally where it stands. So I think I'll end my presentation there, and I'm
Yes,
looking forward
thank you.
to questions
Yes, we have some
and
more
conversation.
questions. There was one about the because you talked about that you also did this trials on humans.
Yeah.
Did you see any? Do you have results yet from that work?
It's not yet published, but we're seeing very clearly that the inflammation is increasing in the humans when they are eating the beef that was fed grain, and it's decreasing when they're eating the beef that was fed grass.
Mm,
Yeah. So it's um we actually there are studies that have proven all of these things individually. There's just never been a study which looks at them all together. And so nothing we've really found so far is surprising. It all kind of already exists in the literature. It just has never been put together into one complete scientific argument.
yeah. We also have a question about that there has been a significant decrease in nutrients since just see, I think my husband just like the modern technique you can turn out the lights.
And the whole house, even when it's off your
Okay, can you use it? Yes, it's very modern. Yeah, sorry. Yeah, so if there has been a significant decrease in nutrients since before, is there previous data to compare to see this shift, like a baseline to calculate the loss?
Yeah, I mean, various governments around the world have been documenting average nutrient levels in food for close to 100 years now. Um,
So
and
it's these average levels that are going down.
the average levels have been going down.
Yeah.
Yes. What our point is, you never get an average carrot or an average steak. You
Mm.
get a carrot that's somewhere in a continuum. And
Yeah.
so instead of thinking, oh boy, all food is bad, poor us, nothing we can do about it, our thought is, there's no average human, there's no average carrot. Everybody
Mm.
sits somewhere in a continuum. If you can begin to choose the things that are better for you and your family, and money is a powerful force in today's world, then you know, that choice on your part of the thing that's good for you will provide a signal to the supply chain to focus on nutrition as opposed to volume. And
Yeah.
then we can have, instead of the peak of the bell curve being in the 20th percentile, the peak of the bell curve can be in the 80th percentile. Almost all the food that's on the shelf could be much more nutritious. It's not
Yeah.
like decrease in nutrients has to happen. It's just that there hasn't been an incentive for um, farmers to focus on nutrition. There's been a focus
Yeah.
on volume because farmers get paid on pounds or kilograms or bushels or tons, not on levels of flavor and nutrition. So
Yeah.
if we can shift the incentive in the supply chain, um, farmers are totally capable of doing a better job. It's really not that hard at all. The basic principles of how to produce healthy food is, you know, it's not hard. Nature does it by itself. It's
Mm.
in many cases the things that we do are counterproductive to that nutritional levels. And so it's really in many cases just going back to supporting the microbiome, and in so doing, we automatically get better levels of nutrition.
Yeah. So what is your next big project? Now you almost finalized the beef study, so what are you going to do next?
Yeah, wheat we have got coming up. Our thought is, we'll take one large um, crop at a time, things that have major ecological footprints and major nutritional footprints. So yeah, wheat is coming up next, and we're designing that study now and trying to do the fundraising. The science is fairly expensive. I mean, not that expensive really, but we're talking many hundreds of thousands of dollars or $1 million per crop. So, because we're doing everything open source and in the commons, as a charitable NGO, um, there's no investment money. So we have to
Mm.
raise the funds through charitable donations to get the science done, and that's
Yeah.
a bit of a hurdle.
If anyone's here listening and want to support this work, please reach out to Dan if you want to support the next study with grains. And I want to ask you as well, Dan, like, why do you think that the whole food industry has focused on volume? Why isn't there more diversification in some focusing on volume, some focusing on nutrient levels? Why is no one doing that today?
Well, I think, the organic movement was in large part about food being more nutritious and permaculture and biodynamics and local. I'm not sure how many of these movements you've had go through Sweden, but I think they've been fairly global. The slow food, the regenerative is a big one right now, agroecology. There have been these streams of sort of alternative ag, it's called, and in many cases they have been you know, having as part of their impulse, we want food to be better. We understand stuff is wrong with our food and we want it to be better. So I think there has been a lot of desire and impetus around that, but um, why has no one actually done this deep science of looking at what the scale of variation is and really connecting it to specific causal factors? Um, is a question that continues to befuddle me. When I figured out that this thing was real, that there was a thing here, and that no one was talking about it, and there didn't seem to be any science and research around it, I was flabbergasted. I was like, how could that possibly be that this thing, which seems really, really important, no one's really meaningfully looked at? And um, I don't know. I can't tell you why. It
And
just
yeah,
it
and do
seems
you
it
think that
seems
this
really
can,
obvious and really important.
yeah, and do you
Yeah.
think that this can be actually picked up by the industry, or do you think that this will keep on being like more small, like yeah, like small movements as you were talking about?
I think it absolutely will be picked up by industry. I think I've certainly have been having conversations with some very large companies about this, and they are very interested in leading. I think basically the concept is, I mean, there's a company called Whole Foods, which is a sort of an upscale retailer, grocery store chain, basically in the States. I think they've got locations in other parts of the world as well. But um, I was talking to someone who worked at Whole Foods maybe 10 or 12 years ago about all this, and I said, you guys have a reputation for having superior quality food. Um, wouldn't you like to help support this research so that people can verify that your stuff is better? And he
Mm,
said, no, not at all. Absolutely not. We know that our stuff is not as good as people think it is. And the last thing we want is people to be able to be testing it.
mm,
But he said, if you are going to work on building these meters and you are going to come out with one, when you're 2 years out from having a meter, please come tell us, because then we'll go to our supply chain and tell that our supply chain that they have 2 years to meet standard, or we'll stop buying from them. So I think it's one of those things where if we have the ability to differentiate, if the consumer has the ability to choose, then that's going to shift the market. And then the big companies are going to jump on that bandwagon and do everything they can to get their supply chain shifted. And that's really the strategy. Our strategy is people make more money by doing the right thing. And so we help them, inspire them to make money, because that's what companies do, is they focus on making money. Um,
mm.
so yeah.
Yeah. So we just hope that it gets picked up by the by more parts of
Well, we
the
have to, I mean, I think what we have to do is we have
thing.
to do the science first. You
Yeah.
can't build a meter to test quality until you've defined quality. And the
Mm,
definition of quality requires millions of dollars. So
mm,
I don't think this is going to happen in any meaningful way until we put the science done. And
yeah.
so that's where we're at right now, is we have to do the science and do it well. And then if we can build the instruments and people can go out to the store and they can begin to see what's good and what's bad, and they can post that on social media, I'm pretty sure the change will happen very fast after that happens.
Mm.
And so,
Yeah, we have a lot of questions coming in chat. Really nice to see everyone being so engaged. We have one about if the nutrient density depends on the soil biology and not on if the farm uses heavy tillage, biodynamic, or regenerative principles. Does that mean that there is no general difference in soil life in these different type of farms? Or did I misunderstand something?
Right. So it looks like it's the levels of life in the soil that correlate with the levels of nutrients in the plant. But you can't say that because the farm is biodynamic, it then therefore has high levels of soil life, because there's many things that are necessary for soil life to be present. For instance, they need air to breathe. And so if your soil is tight, and there's no air, then they can't breathe, then they can't live. And you may be mulching or cover cropping or doing whatever else, but because you didn't have air in the soil, then that, you know, the game over. Water, microbes need water to drink. And so if you have, you have maybe an irrigation line that's going right down the middle of the bed, but it's just really quite dry and powdery in most of the soil, where there's no water, there's no soil life. Where there's no soil life, things don't function. Um,
Mm.
Food is, the microbes need to be fed. They have evolved to eat with, through photosynthetic activity. So plants make sugar in their leaves, and that is injected down into the soil to feed the soil life. If it's wintertime and there's no green leaves growing, then what are the microbes going to eat? If you don't have roots and dead plant material on the ground on the earth's surface, um, the microbes will starve to death. So air, water, food, these are some basic environmental conditions that are necessary for life to function that don't necessarily correlate with being certified organic or being no-till or being biodynamic. Um, Minerals is another really important piece of the puzzle. Cobalt is the center of a compound called B12. Um, if your soil is and 80% of the species of soil life are B12 dependent, which means if you don't have cobalt in your soil, then 80% of the soil life can't exist. So you may be cover cropping, you may be doing no-till, you can be doing all these other things, but if you don't have this critical environmental condition met, then life can't function. So there's a lot of things like that that are quite rudimentary that in many cases we farmers were not taught about, didn't know about, haven't figured out. And we're trying really hard. We're working, we're putting in our hours. It's not a lack of effort. It's just that we don't understand how nature evolved things to work. And then
Mm.
therefore um, we struggle. So
Yeah. So we have a question that's building on that actually. Like if you were to redo the veggie and fruit study, or now when you design the wheat study, will you look at other things than respiration to monitor the soil health or soil life?
absolutely.
Yeah. And what would you add? Because the first study was, as you said, more rudimentary. So what would you look at in the what is important to look at to understand more in the next study?
Yeah, well, I can say with beef, we looked at more than 200 different species of microbes, both in the bacterial and the archaea kingdoms. So I would surmise that that's what we will be doing with wheat as well, is we will be doing a much more intense uh, um, assessment of the microbiome and seeing which species, which families, which genera are present at what concentrations, to see what correlates, what doesn't correlate. But at the moment, we're actually in the process of talking to specialists in wheat to ask them what they think are the things we should be paying attention to, because while we have a sort of a broad base of knowledge, we don't have the deep knowledge. And so one of the foundational things to do before you start a big study like this is to go and talk to the people who are specialists in that field and gain their guidance. So that's where we're at right now is interviewing specialists to have them help us design the study.
Mm. And this one who says that we badly need these kind of studies in Sweden and Europe. And do you know of any initiatives on our side of the Atlantic? except for this one, of course. Inviting you to Tokyo.
Well, I was just in Sweden a couple of weeks ago, meeting at the Axe Foundation, I think it was called. And they were interested at your introduction, Jessica. You were the one who made the introduction to them. They're interested in collaborating. I mean, I'm envisioning this work to be a global project, not to be a American here and European there and Asian there. Like wheat is eaten by people all over the planet. Let's define quality for wheat globally. Let's not think within our political boundaries. Let's think about all humanity and do this once and get it over with. So, as we did with beef, and we tried to have it be global, and we had 4 continents engaged. I'm certainly hoping that wheat will be done that way. I do not want to have the Europeans doing their research and the Americans doing their research and the Chinese and the Indians doing their research. It seems to me to be most intelligent that we all engage this process together. And so, yeah, we're actively working to develop a bunch more European partners and partners all over the world to engage this process. We certainly will be sampling all over the world. We're not just going to be sampling in the US.
Yeah,
Yeah.
so to elaborate a little bit more on that, we at Svensk Kolinlagring really want to work with this issue or these questions because we see it as a really big leverage for working with soil health. Can we also connect it to nutrient density? We think that there's a much more will to pay for those kind of practices that farmers can adapt. And I also think that there's like a whole new world of knowledge when you actually start looking at this, as you say, like cobalt's really important. Why do we not even measure it today? So yeah.
Yeah.
So we really want to work on this, and as you said, we also had X Foundation. They also here listening today, really interested in this topic. But we need more researchers involved. There are not no one at all in Europe today that's focusing on this connection. And I know that there are some researchers here listening, so please start working. And we have 2 spectrometers. We ordered one as soon as we heard of this a few years back. We like, okay, we have to have one. I think you made like 100, and we have 2 of them. So I don't know if there's anyone else in Europe having one, but yeah, so we have 2. So we can just get started basically. So please reach out if you want to.
Anybody who wants to collaborate, I mean,
Yeah,
we're right now looking at EU Horizon grants and other European funding streams and wanting to work with partners across Europe as part of this global endeavor. So absolutely, very much open to any researchers who want to collaborate.
yeah.
Um,
And I
100%.
want to say also, we have used it to measure soil, and we
Yeah.
haven't used it on vegetables yet. So we are still working on getting everything to function.
It has calibrations for soil carbon as well.
Yeah, so that's what we have been using for.
Yeah.
We have another question also, like I take it that the products that had dark green results, like so like the carrots that were dark green in one compound, did they also have dark green in other compounds, or could that be like did you see that?
I get every single time I present this, I get that question, and I really should have the answer, and I don't. Um, uh, it wouldn't take that long. The time I've spent answering this question and saying I don't know the answer, if I just added that all up and I got our statistician to focus on answering that question, that probably could be done. I don't know. I presume if they weren't all in the dark green, they're probably more dark green and light green. But yeah, this is the question. More of all things is not necessarily better. What is good? What it is, where does balance come in? What is nutrition? There's a bunch of deep questions here which we don't really have answers for, and our attempt is to get into that as honestly and openly as possible and engage thoughtful people. And um, yeah, there's a lot of work to do. Like I said, we created this word nutrient density 15, 17 years ago, and there was no word for it back then. And finally, we've gotten enough momentum now and, raised the money and begun doing the research and getting the papers published that there's getting to be some more energy in the space. But this really is the kind of thing that should be done by national governments, should be done by leading researchers, big, international collaborations with credentialed people. As a small educational nonprofit, it really is not fit for us to be the only group leading this effort. Ideally, it would be taken on and done seriously by national governments. I think, the connection to human health, the connection to ecological health, you could argue this, investment from, of government resources into this research um, would be very, very strategic in decreasing costs, right? Decreasing ecological impact from climate, irregularities, decreasing cost from disease. It really, really, I think the opportunities are massive, and we don't need to be fighting anything here. We're not opposed to anything. We're just trying to, use the principles of science to deepen our understanding and you know, facilitate mutual empowerment. Yeah.
Yeah, we have a question also if you also measure fungi. Is it something that you're planning to look at? Or because you talked about microbes, do you
I
also
would
do fungi?
consider fungi to be part of the microbial
Yeah,
community,
yeah,
100%. Not just bacteria, not just RKE. All the
yeah.
all those
All
people
the
down
whole
there that we can't
ecosystem of
see.
microbial community.
Yeah,
Yeah,
100%.
yeah.
Hmm.
There's also one person who asked, well, if where do they find links to your publications?
Bionutrientinstitute.org is the website where we have all of our papers and things.
Yeah, you have a lot of
Right.
reports, but not that many published articles yet. You have
The,
some.
the only paper we formally published in the peer review journal was in Nature Science Communications, and that was to show that the meter that we built and calibrated worked as we said it did, so that this thing could flash lights at food and get readings that were real. As well, we stuck into there some of the variation data on the polyphenols and antioxidants to show how dramatic that variation was. Um,
mm,
but yeah, it's a question of money. Um, I keep saying it over and over again, but like, I would love to have all the money in the world and have done all this, but the reality is I'm just a farmer and I've been trying to do this because it's my life passion, but don't like say, why haven't you done it yet, Dan? I'm like, because I'm just a person. I happen to be running an organization, but we don't have a big line to lots of cash coming blowing into our bank account. And it's a question
mm,
of prioritization of
mm,
resources, staff, time, etc.
yeah.
Um, so
So I ask
help
everyone,
is welcome.
yeah, I ask everyone during the week, like, who is actually responsible for this? Like, who do you think should take the responsibility for this happening with having like I really see the possibility of having like a big paradigm shift where we go from looking at yield to looking at nutrient density. Like that would make such a huge impact, I think, on the whole food value chain food system.
Mhm.
So who do you think is responsible for doing that, and who do you think would be working against such a development?
Well, I think we each can help out in our own way. Whether it's, I've always thought that if we had 100,000 people that were each giving us $5 a month, um, or €5 a month, or I'm not sure how many kroner, maybe 50 kroner a month,
Yeah, yeah,
um, then uh, we could get this done really fast, right? If a few committed citizens of the world who were just willing to tithe a very small amount of their capital into this, 100,000 people out of 8 billion is a very small percentage. Um, so rather than say, oh, this person should do it or that person should do it, I like to say, what can I do? What do I have power? What capacities do I have? So that would be one place to start, is we each could, put our own little bit into the pot and tell our friends and ask them to do it. That would be something that each of us can do. Certainly, we're trying to partner um, with like-minded allies, whether they're researchers in universities or other nonprofits. Certainly, there are, some funding schemes through the government that can be accessed. Um, um, so yeah, for me, I focus on what can I do and who are the relationships what are the relationships I've got that I can act on and begin doing this with. Um, I think it'd be wonderful if, as I said, the National Academy of Sciences in the US and various other sort of well-respected scientific, um, you know, groups would take this on. We had the Human Genome Project that occurred in the 1990s when it was understood that there was an important question that needed to be figured out, and it was bigger than any one research group could do. and it got funded, and it took a few years, but they really figured a lot out. We had a national microbiome initiative here in the States that did something similar. So I think this is a project on that scale that ideally would be done in such a fashion.
Mm. Yeah.
I don't like to think about who would be opposed to it so much. I like to think about who would want to support it.
Mm.
Certainly, it does have the potential to threaten a lot of vested interest. There's a lot of money that is made by people selling fertilizers and pharmaceuticals that potentially their market share would be, or their sales would be dramatically decreased if we succeed. If we succeed, farmers will not be using chemicals. They will not be using fertilizers. The level of medical interventions and disease will be dramatically decreased. So we start there if you want, with groups that might be yeah,
Yeah. So we have another question.
not in support.
Explicit, is it actually hard to get funding for this type of research?
Um, Primarily because of the fact that it's so novel, because no one has ever looked in this space, and there are no published papers because no one has looked. Um, then the way science works is it builds on the canon is built on the foundation of what's already been done. And so
Mm.
um, getting government grants to, be written for this research requires there to be something for the government to say, we want to get grants to further this research. And so it's a chicken and egg kind of a thing. Um, um, I have found it extremely difficult, in part because it's such a global endeavor and it's so multifaceted. If it was just focusing on microbiome, if it was just focusing on carrots, if it was just focusing in one state, that would be one thing. If it was just focusing on soil carbon, if it was just focusing on human health. But the thing is, because it's so multifaceted, because it touches so many pieces, um, it doesn't fit into most funders' portfolios because they generally have a specific focus of what they're funding.
Mm.
So the way we've been most successful is by really is by having personal relationships with well healed people like philanthropists. It's
Yeah.
really been individuals who do have extra money, um, who we can get an introduction to and we can get a personal conversation with, and they can
Mm.
say, wow, this is really important. Okay, I'll write you a $25,000 check or whatever it is.
Yeah.
And yeah, there just aren't really any grants out there being written for this research because no one's done it yet, and there's
Mm.
no papers. So
Another question, if you with the spectrometer, do you intend for the spectrometer to be widely available for consumers, or should farmers each have to have one to measure the quality of their crops? Or how, because you actually not produce, I mean, you are not producing spectrometers, right?
well, yeah, we are a nonprofit, an NGO. We're an educational organization, and we do not want to be a. We're not a company, and we did build a few of these basic ones just to prove principle, but we don't have a vision of being a hardware manufacturing company. Um, what we want to do is we want to get the science done to say this is what quality is, and then let anybody who builds meters calibrate their meters they've already got and sell them with this function. So that's our thought. Um, you know, for me, like I said, the end game is that it will be one of the cameras in your phone.
Mm.
Um, so that you don't need to have a separate meter. And I think
Mm.
that's, as I understand the science, that is entirely possible for that to happen. There might be a couple generations of little handheld meters that we go through for 5 years or something before it gets into the phone as an app. But um, a lot of people have smartphones. Not everybody on the planet has a smartphone, but a lot of people do.
Mm.
And enough people do that we could, figure out pretty readily where this brand's product stands in relation to other brands, basically, because
Yeah.
that's to a large degree what people buy is they have a choice between three different brands. And if you got to know that this one brand was always superior nutritionally, you know, you wouldn't really need to use the meter all the time, or
Mm.
that often even, or that you wouldn't have to have that many people using meters to assess. Um, so
And basically, I think that what Dan is trying to say as well is that the research that they have been doing is open for all. So I think that the um, what you are hoping for, Dan, is really that another entrepreneur will take that and run with it, and just like, okay, so now we will start producing these meters. We have this database. We will build on that database, make it wider, bigger, regionally, adapted, and so on. So this is really a project that I think would be really interesting for a lot of people to have. But you are not the ones. You showed that it's possible, but you are not the ones who are going to produce the meter. But it's also openly available to 3D print on your own. So
The specs, if you want to build one of these yourself, the engineering diagram is how to do it online. It's called open source, which means it can't be locked up. It's freely available for anyone to do. So yeah, if you want to make 1,000 of them and sell them, you can. That's the way we've done it, which is not a business model. It's a philosophical position.
But if
Which
anyone's
maybe is
there
one of our limitations.
listening who wants to like develop this for the consumers, please do.
Well, but you can't build meters to test nutrient density until you've defined it. And that continues to be the hurdle. Until the science is done to define quality, you can't build a meter to test it. Yeah.
So there's also another question about the higher nutrients. Maybe higher nutrients will result in lower quantity and be put against issues with hunger as an argument. Or do you think that the amount of crops will still be the same? I mean, we also heard about it was raining a lot, so the kale became exploded, became really big. Maybe then it was less less nutrient dense than the small one who was in a drought and with super strong taste. Is there what do you think about that?
I got into all this because I'm a farmer, and I was trying to figure out how to make a living, and I struggled with things like pests and disease, and it was through understanding how to work more well with the soil and with nature that I was able to create a situation where my plants were healthier. And when they're healthier, they're not only more nutritious, but they're more productive. So it's kind of a win-win. You don't have to sacrifice quantity for quality. Healthy plants not only produce more food, but they produce higher quality food. Um, and so, yeah, I think in general, people will need to eat less food if there's more nutrients per quantity of food, because it's your body really is looking for nutrition. It's not looking for calories in general. Um, and so we could talk about, hunger globally being able to be reduced. I mean, it's not that we have a lack of food, really. It's just a question of the logistics that causes starvation, really. But yeah, I don't think we should.
I also want to add that there's not a lack of calories in the world. They are not evenly distributed.
They're not evenly distributed.
And we are overeating. There's more people in the world that are obese than that are starving today. So it's really like a political question to make sure that everyone has enough calories as well.
But the thing is, but if they're empty calories, if the
Yeah.
calories you're eating don't have sufficient nutrition in them, you keep eating more calories because your body does need a certain amount of nutrients.
Yeah.
That's the thing. A lot of people are obese because their bodies are telling them, I want more, because the thing that they're eating that they're calling food is actually nutritionally devoid. I call it, I'm not sure you guys have the word junk food in Swedish or how you would say it, but in English, we got a word junk food. And I would say that food is over here and junk is over there. And, don't call this stuff food because it's not, it's junk, and we need to be able to differentiate. And if we simply ate whole food and not processed product, we'd be all dramatically better. I mean, this whole question of like, how good is the carrot? Like, that's sort of the top, that's like the last 10th percentile. The issue really is we're just not eating whole foods like we used to 100 years ago, right? I think that's a really, really important piece of this larger conversation. But
So I think we will finish here. So thank you very much, Dan, for this work that you are doing. Also, some people in the chat has written that they are very impressed with your work. We are very, very happy that you started so that more people can join. And I'm really happy that you said yes to come and spread this knowledge and your insights, so that more people in Sweden can get engaged. And I also just want to finish with, you know, at Svensk Kolinlagring, we're also non-profit, so we have this this gift that we give to all speakers. And sorry, it was in Swedish. I was a little bit in a rush. I didn't have time to
Everyone else is trying to be
to
Swedish.
change it. But it's 1 hectare of transformed farmland in Sweden, 2024. And this is a picture from Amanda Bergman's farm up in Dalarna, with an old variety of cows having rotational grazing in the middle of Sweden. So thank you very much. And I also want to say, if more people want to get engaged, please engage in Sweden. Please get engaged with Svensk Kolinlagring, and you can also buy this transformation package. We also have this as a Christmas gift this year, so soon it's going to be on our webpage if you want to support the work that we are doing, and support, of course, the farmers that are doing the actual work. And that's what we are for, to support the farmers doing transformation on their farms. And we think that nutrient density can help them in the work that they are doing to improve the economy of changing into a different system. So thank you very much for listening. And next on the program, we will have tomorrow, we will be at Kullabergs Vingård the whole day. It's fully booked courses about advanced soil health course, and then plant sap analysis. But there will also be, if people are interested in plant sap analysis and what that is and how we can use it, there will be an open webinar tomorrow afternoon at 3 o'clock with Joel Williams from Canada. And then on Thursday, we will discuss more, crazy ideas of how to work with nutrient density with basic nutrient income, universal basic nutrient income. So it's also coupled with nutrients, and the idea of every people's rights to having nutrient dense food, and how we can use local economies and local currencies as a lever to make sure that we get local produce of nutrient dense rich food. So thank you very much for tonight, and good luck with the election. I hope you will not have chaos spreading in the US in the next coming days. And thank you very much for tonight.
Yeah, thank you very
Bye,
much for
everyone.
having me. Yeah, okay. Good
Bye.
evening. Bye-bye.